Hello everyone, welcome back for the second lecture video on this chapter on human digestive system. Now in the previous lecture video, which you can also watch on my YouTube channel, we have gone through the three main classes of nutrients, namely carbohydrates, proteins, and fats. Okay, we have also gone through what are some examples of food which are rich in these nutrients, as well as what are the uses of these nutrients. Besides that, in the previous video, we have also gone through the purpose, the importance of having a digestive system in humans. But let's go through that once more. So, what is the purpose? What's the importance of digestion in our body? Now, the purpose of digestion, if you recall from the previous video, it is to break down large and also importantly, insoluble food into smaller and soluble nutrients. Now, the purpose of breaking them down into smaller and soluble nutrients is so that the nutrients can be absorbed into the bloodstream. Okay, so this is a very important point. It must be able to be absorbed into the bloodstream so that it can be transported to other parts of the body to be used by the other cells in the body. Now, digestion can actually be divided into two types, physical and chemical digestion. And today we will be learning about the differences between physical and chemical digestion. And then thereafter, we will look at chemical digestion, in particular, the role of enzymes in chemical digestion. Okay, but the first thing to take note of is that in our body, both types of digestion take place. All right, you have both physical and chemical digestion. Okay, so how do they differ? Well, firstly, physical. The word physical digestion is a mechanical breaking up of larger food particles into smaller food particles. Okay, when we say this word mechanical, we are actually thinking of forces. Forces like what? Chewing, grinding, mashing, tearing, cutting, and so on. So these are the actions that we think of when we think of mechanical breakup, which is actually through forces. Okay, so you're mechanically breaking up the large food particles into smaller ones. And the purpose of doing this is actually to increase the surface area to volume ratio so that later on they can undergo faster chemical digestion. So when you chew food into smaller pieces, the individual smaller pieces added together will have a larger surface area to volume ratio, which makes chemical digestion faster. Okay, now because it's physical breakup, there is no change to the chemical composition. You do not form new products. It does not involve enzymes. And finally, the examples, as we have mentioned, physical digestion involves chewing, grinding, mashing. And this is very common in the mouth because you have teeth in the mouth to help chew, grind, and tear up the food. But besides the mouth, if you look at the stomach also has muscular walls, and the muscles can help to mash the food as well. All right, all these are considered physical digestion because it's just a mechanical breakup. There are no new products formed, no enzymes are involved in these actions. However, for chemical digestion, as the name suggests, it's a chemical breakdown. And through this breakdown, the food actually becomes soluble. So you're breaking down insoluble food into soluble nutrients, which of course can be absorbed into the bloodstream. Now, because it's a chemical breakdown, it's a chemical change. As you have learned previously, chemical changes, they form new products. So for instance, proteins, if they undergo chemical digestion, they will form amino acids. Amino acids are new products which are different from proteins. Okay, and chemical digestion involves enzymes, and this is very important. Enzymes are involved in chemical digestion. So where can our enzymes be found in? Now, enzymes can be found in the mouth, in the stomach, as well as in the small intestine. Okay, so in the mouth, you have teeth. Teeth carry out physical digestion. But in the mouth, you also have enzymes, which carry out chemical digestion. And hence, in our third video lecture, we will learn in every single part of the digestive system what is the physical digestion, what is the chemical digestion involved. And you will be expected to describe both the physical and chemical digestion in every part of the digestive system. Okay, but for now, let's learn the differences between physical and chemical digestion, and let's also pay attention to this word enzymes because we are going to be going on to enzymes very shortly. So at this juncture, if you are still unclear about the differences between physical and chemical digestion, I would advise you to pause and rewatch this section of the video. Okay, so now that we have understood the differences between physical and chemical digestion, we are now going to zoom in to this word enzymes. What are the enzymes involved in digestion in our body? So, we need to know. The first thing we need to know is that different enzymes digest different nutrients. For example, if you have carbohydrates such as starch are found in rice, bread, and noodles. So, how do I digest starch? I need enzymes. And the enzyme that digests starch, these enzymes are called carbohydrases. So, as in our previous lecture, we have said anything ending in ASE tells you it's an enzyme. Okay, so carbohydrates is the enzyme which digests carbohydrates. Similarly, protease is the enzyme which digests proteins. Lipase is the enzyme which digests fats. You might be thinking, hey, lipase and fats, what's the relationship? Well, in our previous video lecture, lecture 1, we learned that fats are also called lipids. So fats are called lipids. Therefore, the enzyme that digests lipids is called lipase. Okay? So you need to learn the enzyme name for each nutrient. Carbohydrates digest carbohydrates. Protease digests proteins. Lipase digests fats or lipids. Now, besides learning the enzyme names, you need to also learn the end products of digestion for each nutrient. So carbohydrates. Carbohydrates are digested by carbohydrates to form glucose. Glucose is the end product of digestion of carbohydrates. Okay, now glucose can also be called simple sugar. You also see this word inside your textbook. All right, so you need to learn this sentence. Carbohydrates are digested by carbohydrates to form glucose. Okay, you might want to write down carbohydrates are digested by carbohydrates to form simple sugar or glucose. Similarly, proteins are digested by proteases to form amino acids. So amino acids are the product of digestion of amino acids are the end product of digestion of proteins. And finally, fats are digested by lipases to form fatty acids and glycerol. This is very easily mistaken. You need to know that fats are digested to form two different products. You need to state both products when you are asked for the products of fat digestion. Okay, so this table is a very important table to learn. You will need to learn the enzyme names for each nutrient. You will need to also learn the products of digestion for each nutrient. So again, pause the video to rewatch if you are still unclear about something. Now let's move on to enzymes. So at the start of this video, you learn enzymes are involved in chemical digestion. So chemical digestion is the one that requires enzymes. Now, how are enzymes involved? Enzymes speed up or catalyze chemical reactions. So they make the chemical digestion faster. They catalyze the chemical reactions. Okay, and that's why enzymes are called biological catalysts. Now, at Sec 2, you don't really have to learn how enzymes work. So this is actually enrichment. Okay, so this section on how enzymes work is enrichment and will not be tested in WA. However, I think it's still good for us to have a knowledge and awareness of how enzymes work. So now there's a lot of detail on this page. We will zoom into one section at a time. So let's zoom into the left hand side. How do enzymes work? Now, enzymes, they are unique molecules because they have a unique three-dimensional shape. Now, the unique three-dimensional shape of enzymes includes something called an active site. So I've highlighted in green on the enzyme, that's the shape of the active site. Now, the active site is where the nutrients will bind to the enzyme. The active site is where it binds. And what do you notice? So there's an active site and there's what we call the substrate. Now, the substrate in the case of digestion is your nutrient. The nutrient could be carbohydrate, could be protein, could be fats. Okay, we call these substrates. Substrate just means the molecule that binds to the enzyme. So this is a bit like the lock and the key of your house, right? You put your key inside the lock. The key fits into the lock. So it's similar here. The substrate fits into the active site of the enzyme. So we can say that the enzyme is like the lock and the nutrient substrate that binds, that fits, is the key. The substrate nutrient fits into the lock. All right. So the idea is this is called the lock and key hypothesis. The enzyme has a site where the substrate go in. So the enzyme is the lock and the substrate is the key that goes in and binds to the active site. And you notice, why can the substrate go in? The substrate can go in because the structure of the active site, which I've highlighted in green, the structure of the active site allows the substrate to fit nicely. It's a perfect fit, if you like, in this case. So we say that the structure is complementary to the substrate. Okay? This word complementary means it's a good fit. It fits nicely together. Both the substrate and the active site of the enzyme fits nicely together. And when it fits together, they bind together. When they bind together, the substrate and the enzyme forms the enzyme substrate complex. When they bind together, that's called a complex, and that's where reactions will take place much faster. And when the reaction occurs and finishes, the products dissociate. The products leave. Dissociate means to leave. The products leave from the active site, and the active site of the enzyme is unchanged chemically. This is very important. And this is why you only need very little enzyme to actually digest nutrients. Okay, so the nutrients are chemically changed. The nutrients are digested and broken down. But the enzyme, is the enzyme broken down? No, the enzyme is unchanged. And that's why you don't need so much enzyme. One enzyme can break down many nutrients molecules. Okay? Now, importantly, you need to realize that, hey, the shape of the active site is super important. If the shape of the active site is wrong, is changed, then this whole process wouldn't work. And this is why the active site is the most important part of the enzyme. Without the unique active sites, you cannot speed up the reaction. And this is also why one enzyme only binds to one type of nutrients. Because the nutrients have different shapes, the active sites on their enzymes must also have different shapes. If not, they cannot fit nicely together. And that's why, if you recall the previous section, we learned carbohydrates are digested by carbohydrates. Because carbohydrates have a certain active site with a unique shape that only binds to carbohydrates. Proteins are digested by protease because protease has an active site with a unique shape that only binds to proteins. So if I'm protease, I will only bind to protein. Protease cannot bind to fats. Protease cannot bind to carbohydrates. Protease only bind to protein. That's why protease can only digest proteins. So this is a very important understanding. Okay, the active site shape is so unique, it only binds to one type of nutrient. Protease only binds to protein. Protease can only digest proteins, nothing else. Okay, now this is enrichment, but I think it's good for us to know This helps us explain why do you need different enzymes for digesting different nutrients? Because an enzyme can only bind to one type of nutrient. Okay? Now, the active site shape is so important. We have said so much about the active site. So what can change the active site shape? For instance, if I were to heat up the enzyme, it's so hot, or if I put it into acid, the pH is so extremely low, that will change the active site's shape. So the idea is at high temperatures, or if the pH is too extreme, extremely acidic or extremely alkaline, the enzyme denatures. Now, the meaning of denature is the enzyme will lose the three-dimensional shape. Denature means the enzyme loses its shape. Now, what does this look like? I've given you an example. Over here, you can see on the screen. At first, the enzyme shape has this nice W shape of the active site, right? The W shape of the active site allows it to bind to the substrate, the nutrient. But if I were to heat it up way above 40 degrees Celsius, if I heat it up to a very high temperature, like 100, the shape now changes to a much flatter shape in the active site. The change in the shape of the active site causes it to no longer bind to the substrate. It can no longer fit with the substrate. We say that the active site has changed shape so that it can no longer bind to the substrate. The enzyme is denatured. So denaturing just means a change in the shape of the enzyme and its active site such that it can no longer bind to the substrate. And this denaturing happens when you heat up an enzyme too much, or when you put in extremely acidic or extremely alkaline conditions. This causes the structure of the active site to no longer be complementary, and the substrate cannot bind to the active site to form an enzyme substrate complex. If it cannot bind to form the enzyme substrate complex, then the enzyme doesn't work. The activity is very low. All right. Now, this is enrichment. You will cover this in upper secondary biology if you choose to take bio. Okay. In upper sec bio, you will need to be able to write out this entire explanation from start to the end. And this also gives you a flavor of what upper sec bio will look like. It's not a one word answer. Very often, it's an explanation that must be complete like this. Okay. But this is still important for us to understand because now we understand that, hey, it is dependent on pH and temperature whether the enzymes will work or not. This is important because different parts of our digestive system may have different pH values. So, what works in the mouth may not work in the stomach because the mouth and the stomach may have different pH values. You have learned from primary school, the stomach is very acidic. So, the enzymes in the stomach work well in acid, but they may not work well in the mouth. In the mouth, the pH is actually 7, it's neutral. Okay? So different organs, different parts of the digestive system, different pH, and therefore the enzymes are also different. Okay? Now, finally, in upper sec, you may need to read certain graphs in bio. So shown here, we have 3 different enzymes: pepsin, salivary amylase, and trypsin. You notice that their graphs are different because they are different enzymes. Different enzymes will have different pH at which they work at. Every time you have this kind of graph, we are going to look for the highest activity. Highest activity means best. The enzyme works best. Best is also called optimum. I'm at my optimum means I'm working at my best ability. So the best for pepsin is pH 2. So you notice that pepsin works best, works optimally at pH 2. So the optimum pH of pepsin is pH 2. Now, if you think about pH 2 is acidic. Okay. Now, pepsin is in the digestive system. Which part of the digestive system is acidic? So if you think about it, primary school, you have also learned the stomach is acidic, and therefore pepsin is found in the stomach because pepsin works well in acid in the stomach. All right. Next one, we use the same concept. We always look for the highest point. That's the highest activity. That's the best activity. Optimum. So the optimum of salivary amylase is in the middle between 6 and 8. That's 7, right? So salivary amylase works best at pH 7. Now you have learned in chapter 11, pH 7 is neutral. Which part of your digestive system is neutral? Now pH 7 neutral is found in the mouth. Your mouth is neutral. And that makes sense. Salivary amylase, the word salivary tells you that this enzyme is actually found in the saliva, which is, of course, secreted in the mouth. Okay, so salivary amylase is found in the mouth. And finally, we have trypsin. Again, we look for the highest point. That's the optimum. And then we trace down. Okay, trypsin is about pH 9, 9-ish, okay, about pH 9. The graph is not very clear. Okay, about pH 9. Okay, it's closer to 9. It's not really close to 10. Now, trypsin is pH 9. It works best at pH 9. The optimum pH is pH 9. This is alkaline because pH more than 7 is alkaline. So which part of the digestive system is actually alkaline? Okay, it's the small intestine. Small intestine is where the conditions are alkaline. All right. Now, in our next lecture, we will actually go through each part of the human digestive system, including the pH at every single part of the digestive system. So don't worry too much if you have forgotten whether the stomach is acidic or not. We will go through that again. But what's important right now is to be able to read the graph and get the optimum pH. Okay, so which enzyme functions well only in acidic pH? I think it's quite clear. The enzyme which functions well in acidic pH is pepsin. And finally, we have said pepsin works well in acidic pH, so pepsin is in the stomach because the stomach is acidic. Salivary amylase is found in saliva. Saliva is in the mouth. And finally, trypsin is in the small intestine because the small intestine is actually slightly alkaline. All right. So this is also very helpful. Based on which pH the enzyme works best at, you can actually work out which organ, which part of the digestive system is this enzyme from. Now, I have one more graph on the right, which I would advise that we actually go through together in class. All right, but for now, do take note this section on how enzymes work, the lock and key hypothesis, denaturing is actually in upper sec bio, which also gives you a flavor. If you want to take upper sec bio, you must be willing to write the explanations, which can be quite long, like this. All right, now this is our second video lecture. We have one more video lecture coming up, which is how the different parts of the human digestive system work together. Okay, so please also watch lecture number 3, the different parts of the human digestive system. Okay, and please feel free to rewatch this video if you are still unclear about physical and chemical digestion and enzymes. Okay, thank you so much.